All right, so this is Kate Scow. This is Soil Microbiology, and this is Lecture 3 for Soil Science 111. And the topic of this lecture is kinetics. Kinetics, which are about the rates and what controls the rates of different microbial processes, be they growth, be they metabolism. I want to warn you that I'm surrounded by 3 small dogs, one that barks a lot, he's visiting, and I'm just trying to keep him calm here to my side, and I hope that they don't bark. I don't have any other way of getting rid of them right now, so they're here. So let's wish me luck. Okay, so here's an outline of the lecture today. We're going to look at a little bit about kinetics of metabolism, or the use of substrates by microorganisms. They could be electron donors, acceptors, nutrients. And then we're going to talk about kinetics of growth, which is the replication of individuals. And there's 2 different kinds of cultures of kinetics that describe these 2 types of phenomenon. And then we're going to look a little bit at curves showing these rates over time, and talk about what are some of the particular challenges that are associated with looking at kinetics in soil. I hope this is not a dry lecture for you. There's a lot of really interesting stuff here, and it's a fairly empowering topic because you can get a handle on how to make predictions if you know what are rates of phenomena. Like, it could be how fast does nitrogen become available from an organic source of fertility, like from a manure or compost, or it could be about how long does it take for a particular pollutant to break down in the subsurface, like petroleum around a gas station, or it could be how long does it take for a pesticide to break down in the field after it's sprayed, and can we predict when workers can go back in the field? There's a lot of really important types of application for this information, and let's jump in. Sorry. Okay. All right. Here is a graph that shows you some curves that describe the breakdown of a contaminant, MTBE. It used to be a fuel additive. My lab's done a lot of work on this. And what you see on the y-axis is the concentration of the chemical that is remaining, obviously starting out at 100%, and what happens to it over time, which is on the x-axis. And you see here some different kinds of treatments. This was a lab experiment taking contaminated soil into the lab, and we spiked it with MTBE and followed its disappearance over time. And this is not dissimilar to what we are doing with perchlorate in the lab, in your lab in Soil Science 111. In the case of MTBE, MTBE is the electron donor, and we need to add oxygen. And this is an experiment looking at with and without oxygen. Whereas with the perchlorate, perchlorate is the electron acceptor, and you need to add a donor. But you get the same kinds of curves. So the first important thing to look at here is that sterile control. Remember, we talked about that before in the last lecture, that you need that sterile control, which is the soil that's had the microorganisms killed in it. You remember ways that we did that? It's at the end of the last lecture. Gamma radiation, autoclaving, putting chemicals in. So this sterile control shows that indeed that MTBE did not disappear without there being organisms present. So it didn't just volatilize away, and that was the reason that it looked like it went away. It stayed in there. It needed a living soil for it to break down. And if we look at aerobic soil, so that's aerobic soil that has plenty of oxygen in it. And the first time we added MTBE, it degraded. You can see this curve here showing it disappearing. Then we hit that same soil again with an additional dose of MTBE. So that shows now this curve. And what happened? Is it faster or slower? That's right, it's way faster with the second time it's exposed to MTBE. Why do you think that is? Yeah, it's probably because the organisms that were exposed to MTBE, those in the soil the first time, grew up on that MTBE and were at a higher population density the second time they saw MTBE. Okay, here's another thing that we did. Here is this aerobic soil, first time MTBE was added. And here is soil to which an inoculum of MTBE degraders were added. And you can see that the rate of decomposition was a little bit faster in the presence of the inoculum. But you can also see that it did not require the inoculum for it to degrade, right? Adding an MTBE degrading organism was not necessary in this soil for the MTBE to degrade. The other thing we have here is an anaerobic condition, a flooded soil, like you did in your nitrogen lab, to reduce the oxygen present in the system, and you can see that the rate of decomposition of MTBE is slower. Okay, so here are some kinetics, and there's these interesting stories behind these different curves, and we want to talk about these shapes of these curves and about getting rates off of curves like these. Okay, so kinetics, it's the study of rates and those factors that are controlling the rates of whatever is the phenomenon that you're looking at. Why is this important? It's important everywhere in ecosystem science and ag to be able to have a real deeper and predictive understanding of cycling of nutrients, which are largely controlled by microbes. And I think I already said this at the beginning. It's very important for that. Yep, I already said this point too. So sorry to repeat myself, but these were the points in my introduction. But this warning I did not give to you yet, and I need to give it to you. Be forewarned that these equations that we use to describe microbial processes are way, way, way oversimplifications of what we know is much, much more complicated, especially in soil. So there's a number of assumptions that are required in using these simple kinetic expressions that were really developed for pure cultures in laboratories and flasks. But as long as you understand what is going on, then you can sort of stay on guard and be careful when these things go awry. Surprisingly, often these simple expressions do a pretty good job of describing these rates in soil, which is why we do continue to use them, and we need them because of this number 1 and number 2. But people abuse these equations. For example, somebody who is manufacturing a pesticide that they want to use for a certain kind of application, let's say to spray in strawberries, may want to over-exaggerate how fast that chemical degrades in the soil because they may want to get permission for using it in that particular situation. So there's a lot of number one, direct abuse, but also abuse through just naivete, not understanding what is behind using these different kinetic expressions. So just be aware that these are oversimplifications, and it's important to understand what are the conditions under which these expressions are appropriate. Okay, so we're going to start with substrate utilization. Remember, a substrate is a food source. It can be an electron donor, it can be an electron acceptor like your perchlorate lab, it can be a nutrient source as well. So you all are familiar, I am sure, with the Michaelis-Menten equation or expression, which comes from biochemistry and is really applicable to enzymes, but it has been stolen to use for whole cells, which is what we're using it for in microbiology, because the relationship works pretty well in describing what's going on. So Michaelis-Menten describes the relationship between a rate of substrate utilization and the concentration of the substrate. So here we're making assumptions that all that matter in being able to describe this rate is the substrate concentration. So this picture here on the right, this graph shows you V, the rate of metabolism, and the concentration of the substrate that's being metabolized. And you can see down at zero, the origin, there's no rate, there's no concentration, there is no rate. And then as the concentration of the substrate increases, the rate proportionally increases, but at some point it starts to saturate. It starts to decline and then reaches a maximum rate, which is a parameter called Vmax, which is marked here on the y-axis. So that's the concentration at which, that is the maximum rate of that particular metabolism. And this is for this particular organism that's being described, or this will be applied to, and for this particular substrate under these particular conditions, which are generally ideal conditions where nothing else is limiting for the reaction. We also have another parameter here, which is Km. Km is the concentration, see down here in the x-axis, at which you reach half the maximum rate. So this is just an additional parameter that is pulled off from this relationship between the rate and substrate concentration. This is the Michaelis-Menten relationship. And this equation over here describes this relationship that we're looking at in graphic form. So the V, which is the change in concentration with time, C being the concentration of the substrate, dC dt equals V, equals Vmax, that maximum rate, times C, the concentration of the substrate, divided by Km, this parameter here, plus the concentration. I think this should all be clear from the graph, and everything is pretty self-evident. So this rate, you don't see it here, but is based on a per gram biomass or protein or per cell basis. So to get these parameters, Km and Vmax, requires an experiment where you vary the concentration of the particular substrate. Let's say it's phenol, and you might have a number of bottles that you would replicate with different concentrations of the phenol in it, and you measure a rate, and usually you measure the initial rate of decomposition of that phenol by whatever culture, bacterial culture, that you're looking at who's degrading it. And you would measure this V, this initial rate, at different concentrations. So you might have a number here, a number here, a number here, a number here, and then you can get this relationship from that and be able to measure these parameters Km and Vmax from looking at this relationship. Sorry, there's the dog. All right, there's a couple of interesting things about these two parameters. Km, that have ecological significance. Km is associated with the ability to obtain nutrients at very low concentrations. So oligotrophic organisms generally have low Km values, meaning that they can get to a pretty good rate with respect to that substrate at low concentrations. On the other hand, Vmax, having a high Vmax, is associated with the ability to obtain nutrients at very high concentrations. This would be with eutrophic organisms. Those that can really pig out are ones that have a very, very high Vmax, meaning they can suck up a lot of substrate at a very high rate. Okay, so again, underscoring that there are a lot of assumptions in using Michaelis-Menten expression. The population is limited only by the substrate in the equation. So think of all these other things that the organisms need to use that substrate. So it's not limited by oxygen, not limited by nitrogen, not limited by anything else. And each of those other types of nutrients that the organism need could indeed be described by its own Michaelis-Menten equation. It also assumes only one population is involved in the reaction, and that you're just looking at the behavior of a single group of organisms, and there's no other biological interactions involved. No one's preying on them, no one's competing with them. There's no other drama going on in that flask. There's also no influences of toxins or other kinds of more complicated interactions when an organism has a choice between two substrates. Also, this is extremely important for soil. That substrate is completely and readily available. There is nothing that's going to hold back the access of the organism to that substrate. Remember, this is also from the last lecture. Do you remember some of the phenomena that reduce the availability of substrates? Like solubility, low solubility, adsorption, complexation. So phenomena that keep that substrate from being in solution and easily taken up by organisms. This is a soil problem. Also, the environmental conditions are constant and ideal. They do not change at all over time. Okay, another thing, because we're looking at essentially enzyme kinetics here. Remember, I said we're applying enzyme kinetics to an actual organism. So there can be no net growth of the population involved. Remember, this rate is per cell density or per biomass of organisms, and that cannot change over time. The organisms are not allowed to grow in Michaelis-Menten equations. Okay, that enzyme concentration stays constant. All right, so that Michaelis-Menten equation is the mother of a couple of other equations that can be derived from it that are used extremely commonly in soil, used extremely commonly in environmental biogeochemistry models, and also in biodegradation models. And the type of kinetic expression that's used the most, I would say, is first-order kinetics. So first-order kinetics is a simplified form of that Michaelis-Menten equation that applies when there are very low substrate concentrations present. So, for example, when that concentration of the substrate is very, very small, then that concentration is much, much lower than Km. Remember Km, the concentration at half the maximum rate. So under those conditions, that C, that term in the lower part of this expression is negligible. So you can get rid of it, cross it out, and then you have a constant Vmax over Km, because those are parameters describing those particular conditions. Sorry. And that constant is simplified to this term K1, which is the first order rate constant. All right? And so you've got that rate constant times c. So that's the next line in this derivation, where we can simplify this more full-blown Michaelis-Menten equation to this very simple first-order expression, dc dt equals minus k1c. And then you can modify this equation and into the form that is used in most cases, and you'll be using it too in your kinetics problem set that's part of your perchlorate lab, where going through this derivation here, where Ct, the concentration at any particular time, is equal to that initial concentration times exponent raised to the minus k1t. K1 is that first order rate constant, and t at any one time. Units are negative time, and this equation is also called first order decay or exponential decay. And if you take the natural log of the concentration and plot it over time, you're going to get under best of conditions, a straight line. And the slope of that curve is K1. So you can use this if you don't have more complex nonlinear approaches, which you do using Excel, but you can take the rate constant from this slope. And you don't want to take the rate constant from 2 points. You want to take the rate constant from fitting a line to all the points, and then the best fit of that is what you take as the slope to get K1. Never just interpolate between 2 points. Now, the half-life is another important term that can be derived from this equation expressing first-order kinetics, and the half-life is how long it takes for half the concentration to be removed. And people use this half-life term a lot in talking about pesticides. Like everyone says, the half-life of glyphosate is really fast, it's just a week. They're assuming when they make these kinds of conclusions or statements that it's first-order kinetics going on, which we know applies to situations when the concentration is very low, and it may not always apply. But anyway, that half-life you can solve for it here by setting that concentration to half the initial and solve for how much time that will be, and you get this expression 0.693 divided by the rate constant equals the half-life. So it's pretty simple to be able to calculate the half-life if you know either the rate constant for the first order kinetics, and vice versa. If you know the half-life, you can determine what the first order rate constant is. Okay, so people use first order kinetics a lot to describe things that microbes do. Okay, zero order kinetics is the other simple expression that can be derived from the mother, Michaelis-Menten kinetics, that applies not to low substrate concentrations, but to high substrate concentrations. So when the concentration is very large, in this case, much higher than Km, then what happens is that the Km term is negligible, and so can be crossed out, right? And the equation can be simplified then to V max times concentration divided by concentration, which would be 1. So it simplifies to V equals V max. And we generally use this zero order rate constant, K0, to then describe that rate. And we have so we can rearrange the expression to dc dt equals minus k0. So essentially, the rate at which the substrate is used is a constant rate constant. It's zero order. It doesn't change. It's not dependent on concentration. The units also are negative time, and you can arrange the equation to solve for concentration at any time. Ct equals initial concentration minus that zero order rate constant times time. And also you can get a half-life from it. And, oops. Okay, so there, if you plot the concentration, not the log of the concentration, but just the straight out concentration over time, you get a straight line, and that slope is K0. So that's in contrast, right, to first order, where it's the natural log over time that gives you a straight line, which you get the first order rate constant from. Okay, while we're still back at first order, I want you to note that in this first order equation, there's a rate constant times concentration. So the rate is dependent on what? Concentration. Okay, so here's the rate. The dc dt is the rate, equals the rate constant times the concentration, right? So the rate constant stays the same, but over time that concentration is going to disappear as the organism uses up the substrate. So that rate here, dc dt, is going to get smaller and smaller over time. Don't confuse the rate with the rate constant. Okay, back to zero order. The rate, dc dt, is dependent on what? On the concentration? No. The rate is the rate constant. Does that make sense? Just a linear rate. Every time step, the same amount of substrate is going to be utilized. And remember, it's very, very high substrate concentrations, so the rate is the Vmax. All right, so now if we go back to the Michaelis-Menten, the graph showing the relationship between V and concentration, we can see better about these assumptions. So remember, first order applies to below the Km. When the concentration is below Km, you're in the first order region. And as the concentration, well, let's start with, you start out with all the substrate, as much as there is, and over time, that substrate is utilized and the concentration is less and less and less and less and less. So you can see that your rate declines as the concentration decreases. So in this first order region, that rate, Vmax, or V, the rate is a function of concentration, right? So in opposite of what I just said, the higher the concentration, the higher the rate is going to be. Got it? Okay, and then when you get to the zero order region, you are where that concentration, initial concentration, is way, way higher than Km. And in fact, you're up in this area beyond where that curve is starting to level off, where as the concentration increases, you can see no change in the rate. It's the same rate regardless of the concentration. And remember, that is the Vmax. Okay, so I think it's interesting also to remember, why does this equation saturate? Why does the rate saturate at some point? You probably remember this from biology. You get to the point where the organism cannot take up any more substrate. right? So it's maxed out its ability to metabolize the substrate. And generally, it's what is saturating is the ability of the enzymes to transport, or proteins to transport the substrate into the cell. But essentially, you've gotten to the point where you can offer up a lot more substrate, but it can't handle it anymore. So that's why it finally saturates out like this. And there it will be at its Vmax. Okay, and then what about this region in between? We have this first order region, right, where its rate's proportional to concentration. Zero order region, rate is not proportional to concentration. And we have what I'm calling the Michaelis-Menten region, where the rate is is a function of concentration, but it's not this straight out predictable relationship. And that's where you need to use the full-blown Michaelis-Menten equation to be able to predict the rate. Okay, but people don't like to do that because it's got too many parameters, and it's much easier to use these simplified forms of the Michaelis-Menten equation. And particularly people like to use first order because the shape is kind of forgiving, and we'll describe a lot of different kinds of shapes that people might measure in nature. Okay, and let's just look at these shapes. Okay, so this is not to be confused with this graph that shows the relationship between the rate and the concentration, right? Rate against concentration. Let's now go and look at some shapes of curves which are showing the concentration, for example, of the substrate, 100% or 100 parts per million over time, right? Different curve. And you can see when you plot zero order, you get a straight line. Remember, we showed that already when we looked at the derivation of the equation. It's the same rate over time, just a straight line, whereas the first order has this concave up shape that starts out with the maximum rate, and you can see that you take a tangent to the line to get that rate. If you were just going to use, you know, sort of geometry type approaches, and then over time, what happens to the rate? Yeah, it gets slower and slower and slower, and then finally will level off. Why does it get slower and slower over time? Right, because the concentration of the substrate remaining is less and less and less. So the rate gets slower and slower and slower. This tangent to the curve is going to get less and less and less in its value. And the Michaelis-Menten is going to have some shape that's in between the two. Okay, so first order kinetics are very commonly used by regulators, like the EPA may publish them for different pollutants to use in assessing the environmental impact of these chemicals. And environmental consultants that might work for like gas companies may want to use these rates to predict how long the benzene is going to last in that spill that went into the groundwater from the local gas station. And even though first order rate kinetics have first order rate constants, these are not true constants that you can look up in a book, like what is the solubility of benzene, or what's the vapor phase, or how fast does it vaporize the benzene, or these chemical properties of them are not equivalent. They're not analogous to this biodegradation property, this thing that we call a constant that you could look up in a book. Of course, they're going to be dependent on the conditions that you measure that rate kinetics and derive that rate constant from. So it'll be the environmental, like if you're doing it in like this contaminated subsurface around a gas station, you might take a sample of that into the lab and put it in like a mason jar and replicate it and just follow the disappearance of the benzene in there over time and get a curve. and try to get a rate from it, but it's going to depend on the environmental conditions and what are the kinds of organisms present, what are the properties of the soil, how available is it, and therefore you need to be really careful when you extrapolate these rates from one environment to another. But you will find references that will have tables sometimes where you can look up the first order rate constant for benzene. And it's crazy because it's not a constant. It can't really be used in the same way that it can, that you can use, for example, a solubility term. Okay, and I've already talked about these other aspects. Okay, now we want to switch to kinetics of microbial growth. So in this case, it's not utilization of a substrate, but it's growth of a particular population of organisms. And if we go look at a cartoon showing these different phases, so we're going to focus on bacteria, and this will also apply to archaea. It kind of doesn't apply to fungi because fungi grow filamentously, and we'll talk a little bit about that later, although you may still get these phases. So if we start out here with some logs, this is a log graph, log cell number. We're looking at that change over time. So this is perhaps a flask and it has some bacteria in it and some substrate has been added to it. And we're following what happens to the numbers of organisms in this flask. So you start out with a lag phase often. So a lag phase is a time when there's different kinds of things that can be going on. You can have a lag phase where the organisms don't immediately start to grow if there needs to be enzymes induced for that particular substrate that the organisms are going to be used. You may also find a lag phase because you have nobody in a population who can metabolize the substrate, and they may take some time and undergo some mutations, and you may eventually have some members of the population that can now metabolize the substrate, and then you'll start to see the population grow as a function of that. Or it could be if you have a complex system that there's somebody like protozoa gobbling up the organisms as they're starting to grow, and they are able to keep the population down for a while, but then at some point the population can outgrow the predation pressure and then take off. So this lag phase is a real problem for modelers because you can see it's not necessarily a very systematic or predictable thing going on in determining why there is that lag. And in some cases there will be no lag. Like if the enzymes that are responsible for breaking down the particular substrate are constitutive, then the organisms are going to jump right in and use it, and you may not see a lag phase. A couple of interesting stories about the lag phase is people who study anaerobic degradation of things like chlorinated solvents or other kinds of environmental pollutants. Originally it was very hard to find anaerobic organisms that could degrade these kinds of compounds. And there's a story of a grad student at Michigan State who I think had a culture that he was just incubating for a long time. It took 2 years waiting before that particular sample was able to finally degrade the chlorinated solvent under anaerobic conditions that the student was trying to find an organism from that particular sample. So with patience and waiting out this long lag phase, the student did make a discovery. I think one of the first discoveries of anaerobic degradation of chlorinated solvents. Also in waste treatment systems, there was another, if you have like a flow-through system where in this case it was a pesticide being treated and a company that made the pesticide, there's a waste treatment system like a bioreactor that the waste from making the chemical passes through, and they only have a residence time of a set time for that pesticide to sit in the bioreactor. And what was happening was it was never degrading. It just passed through, and even though it degraded under controlled conditions, it didn't degrade in the bioreactor. And what they found out was that the protozoa that were also present in that reactor were gobbling up the organisms that were starting to grow on the substrate and would eventually use it, but they couldn't get a hold and get their populations up within that time period that particular slug of chemicals stayed in the reactor. Okay. Anyway, and then they finally discovered if they put inhibitors in to get the protozoa out, then they could get the chemical to degrade. All right, long story, sorry. Okay, the second phase is the log phase. So this is the exciting phase where you have, remember, it's log scale. So the organisms are growing exponentially. So this is, again, more relevant to bacteria and archaea. So this logarithmic phase or exponential phase is the active phase of growth. And it's from this particular phase that the rate, the logarithmic growth rate, and the rate constant is calculated. And this period of unimpeded growth where there's nothing limiting the reaction only lasts for so long. If it kept on going, we would have bacteria covering the planet in 6 feet of cells in 27 hours if there was nothing to like stop the madness, stop this growth. Fortunately, things change in the growth conditions that then put an upper limit on how long this active growth phase will last. Okay, the next phase is the stationary phase. So in the stationary phase, there's no net increase or decrease in the cell numbers that grew up on that substrate. And this is a really fascinating phase because a lot of interesting things ecologically happen during it. This is where antibiotics are produced. Endospores, you remember from bacillus, are produced in this stationary phase. Enzymes for lignin degradation, like up from the white rot fungus, are produced in this stage, which is really important and interesting to know if you're trying to promote production of these antibiotics or these enzymes. If you keep them in an active growth phase all the time, they won't do it. They don't do it until they have grown up to a high population, and now they're starting to look for something else. Like, for example, lignin may not be their substrate of choice, but when they run out of another substrate, like the cellulose that might have supported their log phase, then they're going to start to produce enzymes that will break down things that are not as desirable, for example, lignin. That's called the secondary metabolism period. Stationary phase is where secondary metabolism occurs. And then death. Death comes to all of us. And in this phase, you see a decline in the log cell number. And the decline comes from autolysis. That means the organisms lyse, or fall apart, and you're no longer going to see them as cells. And this is when the substrate is gone, and maybe they've gone through a stationary phase. Some organisms don't go through that, but all the food is used up, and also the reserves, like the fat in the cells, is also used up. That's one reason for this. And the second is that you may have actually byproducts or toxic chemicals that are produced that the organisms can no longer live in at the concentrations that are present. This would especially be true in a closed system where you don't have flow through removing all this material, or where you may not have your friends, like your commensalistic relationships, organisms that could help you out by removing things that would be toxic to you. Okay. All right, so let's talk about that. Oh, I wanted to say one more thing too. So this is log cell number, but we could also be looking at biomass of organisms. There's different types of measurements that we could be plotting over time here. It could be spectroscopic measurements of turbidity that reflects cells. So anyway, you can measure different things over time. All right, so let's go back to that logarithmic phase. So bacteria grow by binary fission. So that means an individual cell will grow up to a certain size and then split into two daughters, and then those two daughters will split into two daughters each, and you get this kind of pattern of exponential growth because of this binary fission, where you start out with one cell at the beginning, and what you have is an increasingly faster rate of appearance of new cells over time, till you get to 10 hours, and that one cell is now 1,000,000 cells. And this relationship is shown here. Here's number of cells on two scales, the arithmetic scale, just a straight plot over time, or logarithmic scale, taking the log of it over time. And you can see, so arithmetically, it starts out not too many cells, right? See it here? It's plotting this table of data up here down below is what we're seeing. And you can see it starts out small, just a few organisms, but then they just start to escalate and escalate and escalate. And the rate just gets higher and higher and higher. And if you take the log, you get this straight line. And this relationship is shown here by this equation, dn dt, where n is the number of cells, but remember it can be other things, equals mu, which is that the growth rate constant for that particular organism times the number of cells. And from that equation, then you can go through the same steps that we did with first order kinetics and come up with this equation here that gives you an estimate of what are the density of what's the number of cells at a particular time, equals the initial population density times e to the mu, that growth rate constant, times t, whatever the time is at that point. And that mu is also, just like in first order kinetics, is the slope of this line, the logarithmic plot of number of cells over time. That slope is going to give you that growth rate constant. Just like in first order kinetics, we have our growth rate, dn dt, and we have our rate constant, or growth rate constant. And you can see that that growth rate is a function of the number of cells that are present times that growth rate constant. And you have to be careful with if you're looking at a growth rate kinetics over time, those curves that we were looking at originally that show you the lag, and then the active phase, and then the stationary phase, you have to make sure that you take that growth rate constant from only that active growing phase for the cells. Okay, I think that's it for this. And similar to the first order rate expression, we also have something equivalent to that half-life, but in this case it's the generation time, or the doubling time. How long does it take for the population to double? And this can be derived in the same way, where if that generation time is going to equal, again, that natural log of 2, or 0.693, over the growth rate constant. So a lot of similarities between the first order expression and the logarithmic growth expression. So these values of generation times can be really, really, really low for bacteria who are very rapid growers. That can be as low as, something like 20 minutes for like E. coli. Imagine that they're doubling their population in 20 minutes. But it can be much slower for other bacteria like nitrifiers or some of the pollutant degraders. Anaerobic organisms are going to be much slower. Fungi, if you want to apply these kinds of kinetics to fungi, are going to be much, much slower than most bacteria. And then the mites and the higher organisms, the fauna, are going to be much slower again for this generation time. This rapid generation time of bacteria make them really, really plastic in the sense of being able to respond to environmental conditions, and then because there will always be selective pressure, there's always great potential because you're going through so many generations so fast for new traits to arise in the populations, new abilities to show up. Okay, do fungi really grow by binary fission? They don't really. Most fungi grow filamentously, and actually you can use cubic kinetics, looking at the increase in the biomass of the organisms in 3 dimensions, to do a pretty good job of describing fungi, at least in lab cultures, where they make these beautiful puff balls that just grow bigger and bigger and bigger over time. But in soil, you don't necessarily have them growing that way, and binary fission model that applies to bacteria and archaea the shape does a pretty good job of describing the growth of fungi as well. Okay, so how is growth related to substrate disappearance? I'm just going to mention this here, and you don't need to worry too much about understanding it, but I just wanted to give you a connection, because you know that substrate kinetics that we looked at in the first part of this lecture is related to the growth of those organisms, right? Of course, they've got to grow on something. But you get into a lot more complicated kinetics that is beyond what this course has time to get into. But let's start with this figure in this red box here, where we're following the carbon of a particular substrate. Let's say it's benzene again, and it's being used by a cell, which is going to take up some of that carbon into itself, and that's shown by this assimilation. And 50 to 70% of that carbon is going to go into the cell, and the remainder is going to blow off as CO2, right? Because they have to respire to grow. So some of that initial carbon is not going to end up in the cell. But this assimilation rate is really, really important and goes into calculating this yield, or proportionality term that relates how many cells you end up making from how much substrate. So yield is an important term that you need to relate the substrate utilization to the growth of cells. By the way, someone was asking about greenhouse gases and climate change and using microbes to help to offset perhaps greenhouse gas production, or to help them assimilate CO2 in carbon sequestration. And this assimilation rate, I think I mentioned in my response to that question on Piazza, that fungi have a higher assimilation rate. That means that more of the carbon from a carbon-containing substrate that you put into soil will stay in the actual biomass of fungi than in bacteria. So fungi are going to blow off less CO2 from that substrate that they've taken in. So there's been a lot of interest. Should we enhance fungal growth in soils to be able to increase the carbon sequestration potential of a particular soil, agricultural soil? Okay, sorry for that tangent there, but going back now to what is that relationship between substrate disappearance and growth? And you can see here's our friend, the Michaelis-Menten equation. So we're looking at change in concentration over time. And remember I said it's on a per cell basis or on a biomass basis. So that's shown here by the N, right? Cell density and our growth kinetics. So here's our Michaelis-Menten times our population density times that yield term, that 1 over y, which is that proportionality term related to this box that converts how much of the substrate going in goes into the cell. Okay, so then we get these shapes of these curves, which are of substrate utilization, right? Substrate concentration over time that we talked about at the end of the substrate utilization part of this lecture. Remember, we have first order shape here, and we have a zero order shape there, and then we have that Michaelis-Menten region. Here it's no growth. It's another term for the Michaelis-Menten. Right? So this is under no growth conditions. It's under the examples that we used in this lecture where we didn't allow our cells to grow, remember? So, but let's look at the shapes of the curves, and this is only as far as we're going to go, when you have actually growth of organisms as you're using the substrates. In this case, you don't just have concave up curves or straight lines, like the no growth conditions, but you have concave down curves under the growth conditions where you start out with maybe the rate isn't as high as it will ever be, as it will ultimately be, because the organisms start out in low densities. They haven't grown up yet. And then over time, let's look what happens to that rate, again, of substrate utilization under growth conditions. That rate gets higher and higher and higher and higher and higher and gets really, really fast. And then, of course, it will level off as well. And these are just different shapes of growth-linked kinetics of substrate utilization. And my point here is just that these shapes of disappearance of the substrate are going to look different if you have growth involved. They're going to actually accelerate. Whereas under no growth conditions, the best they can do is stay constant, or they're usually going to decline over time. And this has a lot of implications for predicting how long like a contaminant is going to survive or persist in contaminated soil, or even in how fast nitrogen is going to be delivered from compost. Okay, so these shapes tell you a lot about what's going on, and we aren't going to go any further with this in terms of equations, but you can get into this in a lot more gory detail if you're interested in kinetics. Okay, I think I talked about this at the beginning in that first example. This is an ugly curve that looks a lot more like reality, although the first curve I showed you was not a made-up curve. It was a real curve, set of curves. But this is another experiment, and so I'm just underscoring, and this is relating back to just what we looked at with these growth-linked kinetics, that this purple curve is showing you just a sample of contaminated groundwater sediment that received MTBE, so percent remaining of MTBE the first time, and there was a long lag period, right? And then it degraded over time, and then we gave it another spike of MTBE, and look what happened this time. Pshoo! Immediately degraded. And also with the second time that we hit it again, or the third time we hit it again, it degraded really, really fast, unlike that first time. So this is going to happen over time in contaminated sites where, when the contaminant first hits an area, a region of the soil, it may degrade very, very, very slowly. It may be a long time because of growth of organisms that are not very prevalent to start with, or because of enzyme induction, or maybe because of a mutation. But once they get going, then the rates are going to be much, much faster. Don't look at the sterile control in this experiment. It doesn't look very good. That can happen. Anyway. Okay, so remember we talked about bioavailability, and bioavailability has a big impact on the kinetics of substrate utilization in soil. Remember, this is just directly from the last lecture. The assumption is what is in solution is what can be metabolized. Sorption, solubility, speciation are all going to reduce the availability of a particular substrate that microorganisms could otherwise use. If they didn't have these phenomena in the soil, if they were just sitting in a shaking Erlenmeyer flask, they might just gobble up all of that substrate very, very quickly. But in soil, a lot of it is just not going to be available, and this is very much going to affect the kinetics of utilization of those substrates. And here's a picture that's just showing you an aggregate with some pores inside it. The little black dots are showing the substrate that is in the soil, and you're going to see a little bit of that substrate is dissolved in the soil solution, but a lot of it is sorbed on the surface of that particle, or it may even be hidden inside the aggregate where microbes are too big to get at it, and it's held from them and won't come out unless the concentration outside the aggregates gets very low, and then diffusion might bring them back out. And here we have the bacteria that could use it. Like right here, they're just going to gobble up these hydrocarbon molecules that are in free solution. But the ones that are don't have access to the dissolved form are going to be hanging around the surfaces and waiting for these compounds to desorb or become soluble so that they can utilize them. So what kind of kinetics fits that situation? Well, actually, sometimes first order works. Remember, first order applies to when the substrate concentration is much, much lower than Km. And here you can think of a situation where maybe, let's go call it a hydrocarbon. That's the example that we've got here. So that hydrocarbon may actually be in the soil at a pretty high concentration. It may be like a spill situation, but actually first order doesn't do a bad job of describing it because the concentration of that hydrocarbon that the microorganisms see is actually really, really low because they're only seeing the dissolved phase. Therefore, first order kinetics might do a pretty good job of describing, at least if you're not looking for a high level of precision, it may do a pretty good job of describing the metabolism of that substrate. But what works even better, and you see this kind of kinetics in a lot in the environmental science and people describing pollution degradation, but also in describing the breakdown of plant residues, which have different pools of substrates making them up, some which are really, really readily degradable, and some of them which are in these big polymers that have to be broken down, and they are insoluble, right? And they need to be made available over time. So a double exponential or two first order equations do a very good job of describing this type of phenomenon. So if you look, I can't do it. But anyway, the traditional first order equation would be applying to this available fraction of substrate. This would be like the C0. And then you have a K1 here that's degrading it, for example, to CO2. But the double exponential shows that there's actually a second pool of substrate also present in the soil, which is not available. So, for example, when that hydrocarbon enters in the soil, it's going to undergo these physical and chemical processes that are going to make it sorb to the particles, remember, like in this picture here, or maybe move into small pores through diffusion. And so a large portion of that hydrocarbon is actually going to be present in an unavailable fraction. And then there is this available fraction that will be used really fast by this high first order rate. But there'll be a second first order rate that's going to be quite a bit slower that will be describing the rate at which that compound that is sorbed, tied up on the surface, is desorbed or dissolved or diffuses out and restocks this available fraction of substrate that was utilized by those organisms that are happily using whatever is in solution. So you can write an equation that has these two first order equations to describe the rate overall at which this substrate disappears. So two first orders are often better than one. You'd think, why not 3, 4, 5, 6, 7, 8, 9 different first order equations describing all these different phases or pools of chemicals, but you can't justify the additional parameters. It's just having two first orders does a really good job of describing what's going on, and you don't actually need the additional pools. Okay. Here is a another interesting phenomenon that you see in soil that is related to this availability issue. And this shows you in this case, total petroleum hydrocarbon concentration on the y-axis, starting with the initial concentration of that over time in two soils. Soil A is like a soil with not much organic matter, could be a really sandy soil, and you can see that it degrades pretty rapidly over time and very quickly gets below this concentration that regulators have determined is a safe concentration in soil. Therefore, if you can get that contaminated soil, which is initially at a high concentration of petroleum, down to below this level, then it's deemed treated and safe, and you no longer have to worry about remediating it. So in the soil that doesn't have a lot of organic matter, not a lot of sorption, it will break down pretty fast, pretty straightforward. Regulators will stamp that site with a stamp of approval, carry on. Whereas if you have a soil with a high organic matter content, then as we showed here in this equation, a large portion of that petroleum is going to be unavailable. It's going to be in this slower pool that will only slowly be released into the available fraction as the organisms are breaking down what is there, controlled by this rate of desorption or dissolution or diffusion. And in that case, this is actually a double exponential shape here. You have a initial high rate, but then it goes into a very, very slow second phase of degradation as controlled by those slow desorption or dissolution rates. And it may be that it'll take forever, or maybe you'll never get to below the regulatory concentration, the concentration that's seen to be safe, and it'll persist for a long, long time. And this happens quite a lot, and it brings up a lot of discussion by regulators that if it's so tied up in the organic matter and it's taking so long to degrade, does it really matter? Is it really there? If microbes can't get at it, is it going to be a problem for plants? Or is it going to be a problem for worms? Or is it going to even come off and flow down into groundwater? And there's a lot of research that's been done on this and coming up with, should we have site-specific acceptable levels of contamination? That is a function of, for example, the fact that a soil has a really high organic matter content, and you may never be able to get the concentration below a level that regulators have deemed to be a safe level. Okay, we'll discuss that a little bit more when we talk about kinetics at the end, but you can see soil is pretty fascinating because of this availability issue. So this is just a cheat sheet for remembering the types of kinetics that we're looking at. So remember, there's growth and substrate utilization, and we have the Michaelis-Menten equation that's describing substrate utilization. There's actually a Monod equation here, but this is not required for you to grasp, but it looks a lot like the Michaelis-Menten equation. But that's the mother equation that we would use to describe growth. But remember, we aren't talking about it in this class. We're just looking at a special case, which is logarithmic growth, and for growth. And then for substrate utilization, the special cases, or the simplified forms of this Michaelis-Menten mother equation, are first order kinetics and zero order kinetics. Note the shocking similarity between logarithmic growth and first order kinetics. So you can see, I don't know why there's a line underneath the dCdt here, but the only difference in the forms of these equations are what? That negative sign, right? So the first order rate constant is negative because you're looking at a decay or a disappearance over time. With growth, it's a positive, and because we're looking at accumulation or increase in cells over time. Similarly, these forms are for solving for cells or concentration over time have also the same form. They both have this amount remaining at a particular time is the initial amount, be it cells, be it substrate, times E to the rate constant times T. And here again, so we have a positive rate constant here for growth and a negative rate constant for first order substrate utilization. And similarly, we can use the same expression to solve, which is a natural log of 2 over the first order rate constant, or the growth rate constant, giving us the half-life or the generation time. Okay, so you just need to understand these general forms of these equations and just keep track of whether you're talking about growth or first order decay, but they're basically the same forms, just differing in those terms. Okay, this is appendix, Monod equation. That's old school linearization of Michaelis-Menten equation. Some of you may still do the Lineweaver-Burke transformation, these ways that people manipulated equations to be able to get straight lines because we didn't have nonlinear techniques to be able to get rate constants from these relationships between concentration and rates. Now it's much more straightforward. Excel can just determine those for you without having to linearize these equations. So this is just for your interest. And okay, we stop there. Thank you. Sorry for a little bit of distraction with these dogs, and but that's the end of this kinetics lecture, and hopefully this will be of help for you in doing your perchlorate lab, where you're going to take the data that you have collected, but also that the TA and SRA have collected over time to be able to give you a nice curve so that you can figure out the kinetics of perchlorate degradation from your lab and be able to answer some applied questions that someone might have to answer if they were dealing with a cleanup situation and trying to predict how long the perchlorate's going to persist. So hopefully this will be of interest to you, but remember, you ecosystem types and the ag people, these same kinds of kinetics are also important for the phenomena that you might be looking at, like rate of nutrient release or methane production or consumption, whatever it is you're looking at. These do apply. But again, beware of what you're doing, and always be skeptical and think about the assumptions that you're making and how you can't go too far with this. All right, that's the end.